So the title is ambitious because we have seen that we are falling behind in CAFA. So we are trying to find new ways to apply our method. I'm Giuseppe, I'm from the Biocomputing group at the University of Bologna. This is our method. It was first published in 2009. It has not changed in the last 8 years. We start from the whole UniProt. We align the sequences, each pair of sequences, and we put sequences that share at least 40% sequence identity over 90% of the sequence alignment in a cluster, the same cluster. And then we perform a statistically validated a statistical validation of the annotation inside the cluster. So the idea is when a new sequence enters a cluster by following these rules, this sequence inherits the statistically validated annotation. We also provide HMM models for the cluster from the structures inside the cluster. This was very good in the first CAFA. In the second CAFA, maybe. And then in the bar 3.0, we added more information to move out of just predicting the function. And we add cross-cluster interaction, so the new sequence may inherit some idea of interaction with something else, and we added new functionalities for searches. Before we had just, you have the sequence, you get the annotation. Now you can look for what is having that Go term, which PDB is in that cluster, and so on. This is just a brief statistics. The most interesting part is the inherited. The inherited percentage is the percentage of sequences that lacked that specific annotation and is getting a new one. Only the gray part is the one that is missing any annotation. This was a comparison that we, for this version of BAR, we cannot participate in CAFA 2 because it was not ready. And then we simulated it on the CAFA 2 data. And if you see, the most important part is the comparison between the blue bar, which is the current version, and the green bar, which was the version that participated in CAFA 2. And the molecular function number is less than last year, because last year was wrong, and this one is the correct one. But we still are behind the best performing methods. So, how we did this year? I'm not commenting it, because I just checked the benchmark and how we fared. I think we did very well on just some organism like E. coli. I think we did very well, but I'm waiting for the actual data. And another organism we did very bad, I think, in human. So we are out of the best performing methods. But this is a chance. CAFA is always a chance to find new ideas in how to improve the method. And for example, I'm going to check if we are predicting Go terms that are deeper than the actual one. So we are getting false positive because we are more specific, or we are getting false positive because we are out of that branch. And we are integrating also our predictors for subcellular localization to improve the method. And I think I'm done. And I want to thank all the group and Elixir Italy for funding my position. Thank you.